And then we'll be able to push the button on the other side. Okay, I'll get us started. Good morning, everybody. Welcome to this week's edition of Micro Seminar. It's my pleasure to introduce to you Elhanan Tzipilevich, who is a postdoc at Duke University in Philip Benfey's lab. He's been there for the last couple years. Prior to that, he did his PhD at Hebrew University in Jerusalem under the advisement of Sigal Ben Yoda. He has a PhD in microbiology and specifically in phage bacterial interactions. And we're going to be hearing about some of his work on that today. And so without any further ado, I'll turn it over. Take it away.
Okay, good morning, everybody. Today I'm going to present you the work that I did at the end of my PhD under the supervision of Professor Sigal Ben Yehuda at the Hebrew University of Jerusalem about plaque formation dynamics and what it tells us about phage bacteria interaction. So phages are viruses that infect and kill bacteria, and phage research have a major impact on many important fields like microbial-based bioindustry, where phages are the enemies that can kill a whole batch of bacteria as antibiotic alternative to kill pathogenic bacteria, or for the biotechnology industry, where many tools that you all know, like restriction modification enzyme and CRISPR, were found during a research on phage bacteria interaction. And this is a phage life cycle, which I will go over briefly. Phages float in the environment until they encounter the host bacteria. They attach to the specific receptor on the host to inject their DNA. And then, in some cases, the DNA can integrate it into the bacterial chromosome and replicate together with the bacteria, what's called a lysogenic cycle. And upon sensing some environmental signal, it exists from the host DNA and go into the lytic cycle. Other phages are strictly lytic and go directly to the lytic cycle. In the lytic cycle, the phage DNA is replicated, transcribed, and translated, and mature phages form. And finally, the phage secrete enzymes that lyse the host cell and release the phages for another cycle. And these are phage plaques on an LB plates. Phage plaques are those tiny holes formed on a lawn of sensitive bacteria when phage replicate and spread over the bacterial lawn and lyse the cells. And although phage plaques are known for more than a century from the early times of phage research, and they are utilized to indicate the presence of a phage or to enumerate the number of phages, the dynamic of plaque formation is not completely understood. So to address plaque formation dynamic, I utilized as the model system the gram-positive soil bacterium Bacillus subtilis. And I utilized a strictly lytic phage from the Cephuviridae family called SPP1. It's harbor a long non-contractile tail, and also sometimes another phage called Phi29 from the Podoviridae family with a short tail. And both phages bind the cell wall associated sugar polymers called cell wall teichoic acids as a receptor in order to inject their DNA into the cell. So first, I monitor plaque formation dynamic by SPP1 on a lawn of M. cherry expressing Bacillus subtilis, and you can see here the initial stages of the plaque after around 4 hours, and it's over time the plaque grows as more and more bacteria are lysing, but surprisingly, after some time the process is reserved and the plaque begin to shrink back, and the final plaque size after 14 hours was much smaller than the maximal size that the plaque reached during the formation process. This is also plaque formation by GFP labeled phages, and you can see here the same phenomenon of plaque growing and shrinking. And as you can see here, also during shrinking, there is a high concentration of phages inside the plaque. And after we observe this phenomenon in high resolution, we found that we can also observe it on normal plates. When we monitor them over time, we see the plaque growing and then shrinking back. And this is quantification of this phenomenon. And at the final time point after overnight, the time we regularly look on the plaques, their size is around 50% of the maximal size that they reach during the process. And this is with SPP1 infection, but similar phenomenon we found with Phe29 infection. So as I showed you, there is high concentration of phages on the plaque periphery, but anyway, bacteria can grow inside the plaque. And interestingly, when we took this bacteria from the plaque edge, we found that they are still phage sensitive. So this raises an obvious question of how can sensitive cell grow in the presence of phages? And this brought us in mind the idea of phage tolerance states. And more specifically, the hypothesis is that phage at plaque periphery sends some danger signal coming from the infected cell at the plaque center and activate kind of tolerance response, making them more tolerant to phage infection. An obvious candidate for sensing such signal and activating such response are extracellular responsive sigma factor or ECF sigma factor. ECF sigma factor are sigma factors that respond to stress coming from outside the cell and activate stress responsive genes. And Bacillus have 7 such sigma factors, and to explore their effect on plaque size, we deleted each one of them. You can see here normal distribution of plaque size on wild type bacteria and no change in 6 out of 7 ECF sigma factors. But when we knocked out sigma X, we found much larger plaque size. And to specifically explore what is the defect in plaque size in sigma X mutant cell, we explore plaque formation dynamic, and we found that sigma X mutant cell are specifically perturbed in the plaque constriction stage upon infection with SPP1 and similarly with Phe29. So this suggests that sigma X is necessary for efficient plaque constriction. To further explore the role of sigma X in plaque constriction, we mix together M-cherry labeled wild type cells with GFP labeled sigma X mutant cells. We infect both with the SPP1 and monitor plaque formation dynamic at high resolution. At the beginning, the plaque grow and both cells are lysing, but when the plaque shrinks back, we found that GFP labeled sigma X mutant cells are largely excluded from the constriction area. Again, suggesting that sigma X is necessary for efficient plaque constriction. Next, we monitored sigma X expression dynamic utilizing sigma X promoter fused to GFP. And indeed, we found that sigma X expression increases specifically during the constriction phase. To further explore sigma X dynamic in non-infected cell following lysis of their neighbors, we mixed together phage-sensitive cells labeled with M-cherry with phage-resistant cells harboring an extra copy of sigma X fused to GFP. Those cells are phage-resistant, so they cannot themselves be infected by phages. However, they can sense the lysis of their neighbor-sensitive cells. And indeed, after the lysis of the sensitive cells, we found increase in the expression of sigma X and also a major change in localization. And the localization and level went back to normal after complete lysis of the whole community of sensitive cells. Interestingly, this change in localization follows a known dynamic of ECF sigma factor. At the beginning, it's in membrane-bound inactive state. Later, when it sends a stress signal, it changes localization to a nucleoid localization to direct gene expression. And finally, after the stress is removed, it's rapidly returned back to a membrane-bound inactive state. So this suggests that SigX is activated in non-infected cells upon lysis of their neighbors, similarly to what hypothetically happened inside the plaque. To further explore the role of SigX in phage tolerance, we use the SigX expression from an IPTG-inducible promoter. And indeed, overexpression of SigX confer clear phage tolerance. This is a lysis of wild-type culture or culture without induction after phage addition, but addition of IPTG to induce SigX confer phage tolerance upon SPP1 infection and similarly upon Phe29 infection. Further, we did a time-lapse microscopy where we mixed together wild-type cells with M-cherry labeled cells harboring an inducible SigX and infect both with GFP-labeled phages. And we found that at the beginning, all the wild-type bacteria got infected, but the SigX overexpressing bacteria remained intact and got infected only much later, and also mostly remain intact even after lysis of most of the sensitive wild-type bacteria. So this SigX inducible system enables us to ask at what specific infection stage the SigX perturb phage infection. So the first stage of phage infection, as I showed you at the beginning, is adsorption to the cell. And we found that adsorption to the cell is normal even when we overexpress SigX. The next stage is DNA entry into the cell, and we monitor DNA entry using a FROS system. FROS is fluorescent repressor operator system. The bacteria harbor a repressor, specifically LAC-I, fused to a fluorescent protein. And upon phage infection, the LAC-I binds to a LAC operator that was introduced into the phage genome and forms this foci. And monitoring this foci revealed that DNA entry into the cell was perturbed in cells overexpressing SigX. And almost no foci formed. But this SigX inducible system enabled us to further refine the specific step that is perturbed. The stages between binding and DNA ejection can be further divided into sub-stages. At the beginning, the phage bind reversibly to the cell, and then the binding change from reversible to an irreversible binding, and finally DNA is injected. And the difference between reversible and irreversible binding can be monitored by diluting the cell culture. And indeed, we redo the binding assay, but at this time, we diluted the culture after phage binding before we monitored the number of bound phages. And we found that when we activated SigX, dilution significantly reduced the number of bound phages, suggesting that SigX perturb phage infections specifically at the transition from reversible to an irreversible binding. So I want to summarize up to this point. I show you at the beginning that plaque formation follow an expansion stage and then a constriction stage. And this constriction stage is due to cell growth into the plaque. And I hope I was able to convince you that constriction is directed at least in part by activation of SigX, and SigX expression confers phage tolerance by perturbs the transition of the phages from a reversible into an irreversible adsorption. So this raises a question: what happened to the phages that were able to sneak into the cell? Because as I showed you, it's not a perfect protection. And to answer this question, we did again this time-lapse microscopy where we mixed together wild-type cell with M-cherry labeled SigX inducible cells and infect them with GFP phages. But this time we let the phages enough time to inject their DNA before we activated SigX. And indeed, you can see that both cells got infected in the same time. Both have red and dark cells have GFP inside. However, although they both infected at the same times, cells overexpressing sigma X lyse much later and remain intact after the lysis of the sensitive wild type cells. And also, when we monitor the OD of the culture, here we added the phages at 1 hour and either induce sigma X by adding IPTG after half an hour or just right after phage addition. And you can see here that even when we added IPTG after phage, they are still protected from lysis. Then we monitor the burst size. Burst size is the number of phages formed inside the cells. And we found that when we added IPTG before phage addition, almost no phages form inside the cell. However, when we added IPTG right after the phages, the number of phages the number of phages formed inside the cell was completely the same as wild type cell, but although there is the same number of phages, they are still protected from lysis. So it looked like phages develop normally when we induce sigma X after phage invasion. However, cell lysis was still perturbed. So to directly explore the possibility that sigma X can protect the cell from lysis, we utilize a purified SPP1 lysis, and you can see here that overexpression of sigma X confer resistance to purified SPP1 lysis, and interestingly, sigma X knockout enhances sensitivity of the cell to purified lysis as judged by minimal inhibitory concentration. And also here, you can see that 20 minutes after addition of the purified lysine, the whole wild type culture was lysed. However, even after 1 hour of addition of purified lysis into cell overexpressing sigma X, many cells remain intact. So sigma X can also protect the cells from phage lysis. But sigma X is a sigma factor, and it's induced the expression of 18 genes and operon. So the next question is what specifically sigma X target is necessary for this phage tolerance response? And to explore this question, we did a kind of a screen where we knocked out 14 out of the 18 targets of sigma X on the background of sigma X overexpression, and looked for phage tolerance. And you can see that most of them remain phage tolerance upon phage infection. However, one mutation in DLTA, which disrupt a whole operon called DLT operon, abolish the tolerance phenotype. And here again, you can see here the lysis of the culture, and upon induction of sigma X in red, there is a phage tolerance. However, when we induce sigma X in cell with mutation in DLTA, the phage tolerance was largely abolished. And we found that DLTA deletion can perturb all the phenotype of sigma X overexpression. Here is a time-lapse microscopy where we mix together M-cherry labeled wild type cells with non-labeled cells harboring an inducible copy of sigma X together with mutation in DLTA, and in fact both in GFP label SPP1. And we found that both got infected at the same time and also lysed at the same time, and no tolerance response was observed. And also the irreversible binding, when we express sigma X on the background of DLTL mutant and wash and dilute the culture, there is no release of phages. So I showed you the DLT operon is necessary for sigma X induced phage tolerance. And interestingly, DLT operon is known to catalyze the attachment of D-alanine molecules to the cell wall teichoic acid, the same teichoic acid that, as I mentioned at the beginning, utilizes a phage receptor. Further corroborating the role of DLTA in plaque constriction came from the fact that DLTA essentially recapitulates all the phenotype of SigX mutant, it have larger plaques. It is also perturbing plaque constriction. And also when we mix together GFP labeled wild type cell with M-cherry labeled DLTA knockout cell and monitored plaque formation, we found that M-cherry labeled DLTA mutant cells are completely excluded from the constriction zone. And finally, when we monitor DLTA expression by fusing DLTA into a YFP fluorescent protein, we found that DLTA is specifically expressed during the constriction in the area around the plaque. So to summarize, I showed you today that monitoring plaque formation dynamics reveal a previously unknown phase of plaque constriction. This plaque constriction came from phage-sensitive bacteria that were able to grow into the plaque through transient acquisition of phage tolerance. And this phage tolerance was directed, at least in part, through SigX activation in non-infected cells. This SigX activates a fail-safe mechanism that protects the cells from outside by blocking irreversible blinding. And also, if phages are able to invade into the cell, it protects the cell from inside and perturb lysis of the cells. And SigX confers this phage tolerance through activation of DLT operons that modified the cell wall teichoic acid with D-alanine molecule. And just before I finish, I want to raise an interesting question. I just briefly mentioned at the beginning that SigX is a stress response sigma factor. And it is known that SigX is activated by environmental stressors, like, for example, heat stress or salinity stress. So this raises an interesting question of what will be the dynamic of phage infection during such stresses. And to begin to address this question, we monitor plaque formation during heat stress. You can see here that SigX is activated when we transfer the cell from 37 degrees to 48 degrees. And when we monitored plaque size, we found that at 48 degrees, bacteria form, the phages form a very tiny plaques upon infection with SPP1 or Phe29. And these tiny plaques will depend on activation of SigX, because when we deleted SigX, the plaques return to be a larger plaques. And this is a quantification. You can see here that upon transition of 48 degrees, the plaque size of Y-type cells highly decreased. However, the plaque size of SigX mutant cells only decreased a little bit with SPP1 infection and Phe29 infection. So this shows that activation of SigX when one stress, like heat stress, for example, can confer cross-resistance to another stress, to phage infection. And this raises an interesting possibility that in stressful environment like nature, where bacteria are coping with stress all the time, they will be permanently in a phage tolerance state. And phage-bacteria interaction will be completely different in such environment than what we regularly see in the lab. And with this, I want to finish and thanks my PhD mentor, Professor Sigal Ben-Yehuda, and also thanks Osher Fiaxel, a PhD student at Sigal Ben-Yehuda Lab, which currently finalized this work for publication. I also want to thank the Hebrew University and Hadassah Medical School for hosting me for 5 years during my PhD. And finally, I really want to thank the International Society of Microbial Ecology for the opportunity to present my work in this wonderful micro seminar. And thank you for listening. And I will be happy to take questions.
Okay, thank you. That was very interesting. You've got a wonderful system for exploring these dynamics. It was impressive to see how with all the gene knockouts you just had one that matched the wild type. That was really cool. I'm gonna ask a couple questions myself and see if any come in through the chat. we are in a little bit of a time delay, so I'll let questions come in that way. But I wanted to ask: is the deletion in the DLT operon, was that a deletion of an entire locus, or was that just a deletion of a single gene within that operon?
So it was a deletion of the first gene of the operon, which probably should disrupt the whole operon. First, it's an essential gene for the function of the operon, and also it is the operon that all the genes are expressed together, so disruption in the middle, but it's only the first gene.
Okay, and how many genes, just out of curiosity, are in there?
6 genes.
Okay, cool. Okay, we have a question coming in the chat from Daniel Schwartz. Very interesting, thanks. Question: is there a cost to this tolerance? Why not be tolerant all the time?
Yeah, so SigX activation is costly. We see a growth delay when we artificially overexpress SigX. Although it's a higher expression than what probably happened normally, but it's hints that probably there is a cost for activation.
Okay. I have another question along kind of well, I don't know if it's related to that, but I'm really curious about this phenotype that you found where you have the phage that are able to replicate inside the cell, but the culture was able to survive, you know, at a population level from mass lysis, even though the phage had replicated. And I'm wondering if you can, you know, propose or link the mechanism for, you know, the DLT operon to why the cultures aren't dying from lysis in that one particular phenotype where you added the IPTG late.
Yeah, so I don't know for sure, but I can guess it was shown previously as DLT is important for antibiotic resistance. It shows that DLT overexpression changed the membrane rigidity. So it can be that this change in membrane rigidity somehow perturbs the lysis. There are also other possible mechanisms because the lysis is not one hit event. It needs to lyse the cell at many places. And it was shown that lysins need to bind, they need to bind to the cell wall. And if you delete or perturb the binding abilities, they do not lyse well. So it can be change in membrane rigidity, it can be change in the binding ability of the lysins. Those are possible mechanisms to explain this delayed lysis.
Interesting. Is that something that you're going to follow up on, or maybe another way to ask that is where do you go next with this line of research?
Yes, yeah, so I'm currently during my postdoc continue to do phage bacterial interaction and mainly concentrate on what I proposed at the end of how environmental condition affect this phage bacterial interaction. I'm doing it on plant roots and also see that it's different from phage bacterial interaction on LB culture, suggesting that such environmental conditions that change in expression can also indirectly affect phage bacterial interaction.
All right, very interesting. Okay, well, thank you so much for your contribution. We're really glad to have this. We've had a growing body of phage talks, so we're really pleased to have you. There's no additional questions at this time, but check back into the chat, and some people may, or not the chat, but the comments below. Some people may actually ask questions later. But thank you again, Dr. Tzipilevich. Very grateful for your contribution.
Thank you very much.